We're happy to have with us today Dr. Brion Maher. Dr. Brion Maher, he is a professor and co-director of the NIDA Epidemiology Training Program in the Department of Mental Health at the Johns Hopkins Bloomberg School of Public Health. He received a PhD in human genetics from the University of Pittsburgh and specialized in quantitative genetics. His research focused on the genetic risk and epigenetic correlates of complex human disease, including psychiatry and substance of use disorders. Well, thank you for being with us today, and I'll pass it to you.
Yeah, thank you. So you can see the title of my talk. It's Genetics and Epigenetics Applied to Substance Use Disorders in Humans. I'm always torn between how to describe the relationship between epigenetics and substance use because as I study it, it's frequently a consequence of and not just and maybe not a causal correlate. So a bit of an outline of what I'll talk about. First, because I know many of you may be unfamiliar with how substance use disorders are defined in individuals and in the population. I'll discuss that. And then I'll discuss a little bit about the current state of the field over the last several decades on the state of what we know about genetic influences on both substance use and substance use disorders. And then I'll talk a bit about epigenetics and substance use, just sort of generally how we're thinking about that and some of the recent work that I've been part of. And then I'm giving you the conclusions up front, but the substance use disorder diagnosis probably doesn't adequately capture risk across the lifespan, and a continuous liability approach may be better. And I'm sure that many of you, with regard to have encountered this with traits that frequently get dichotomized, that may be better explained as a continuum. So over the last several decades, there have been publications of what is called the Diagnostic and Statistical Manual of the American Psychiatric Association. And in that Diagnostic and Statistical Manual, by version, there have been different symptoms and different thresholds for defining substance use problems. And prior to the most recent version, which is DSM-5, DSM-4 separately diagnosed abuse and dependence. And over time, the concept of a use disorder became was accepted as a better approach to describing substance use problems. And so the concept of both abuse and dependence are no longer used clinically. But you can see that essentially all of the items from the DSM-4 got carried forward, with the exception of legal problems, because that's defining what is a clinical diagnosis based on someone's experience with law enforcement. So that's been dropped. But craving, which is probably an important descriptor of use problems or use disorder, has been added. And so anyone who has two or more of these symptoms would meet diagnostic criteria for a particular substance. So these questions are asked of each substance, and you can see the list of substances below. 2 or 3 symptoms would indicate a mild substance use disorder, 4 or 5 would indicate moderate use disorder, and 6 or more is a severe substance use disorder. And as I mentioned, these are substance specific. But, the question is a substance use disorder diagnosis of an actual reflection of reality, or is it, does a 2 or a 3 or a 4 really mean anything with regard to, with respect to individual comparisons? And so in DSM-5, considering that you need greater than 2 or more, you need 2 or more out of 11 symptoms, there are 2,036 possible combinations. And so these criteria are selected based on evidence. It's a very heterogeneous phenotypic definition. And the former director of the NIMH, National Institute on Mental Health at the NIH, said, unlike our definitions for ischemic heart disease, lymphoma, or AIDS, the DSM diagnoses are based on the consensus about clusters of clinical symptoms, not any objective laboratory measure. And so we're then stuck with studying what are diagnoses that are not based on any one clear-cut threshold or one clear-cut underlying physiological process. And then complicating it even further is that substance use occurs in stages, with someone first initiating the use of a substance, say, alcohol or opioids, and then continued use after that, eventual abuse, meaning overuse of the substance more than someone anticipated or planned. Ultimately, someone can become physically dependent or physiologically dependent or psychologically dependent on that substance. And frequently in the career of a substance user, they'll ultimately cease use. And so in thinking about this as a disease or a disorder where we want to study the genetic contribution to the risk, the epigenetic contribution to the risk, and the epigenetic consequences of that, we really have to think about what part of this process we're actually studying. An example I like to give when talking about substance use disorders is PKU, phenylketonuria, which is an autosomal recessive disorder caused by mutations in phenylalanine hydroxylase, and reduced PAH activity leads to accumulation of phenylketone, and this is a neurotoxin. And the phenotype prior to newborn screening for PKU was intellectual disability and seizures, and the treatment is removal from removal of phenylalanine from the diet. And in this graph here, environment E4 describes the current environment where we have ubiquitous testing at birth for the presence of high phenylalanine via heel stick in newborn babies. And once we modify the environment, the phenotype is identical for non-PKU folks versus those with the PKU genotype. And so the question then becomes, is PKU a genetic disorder, or is it a disorder of the environment? And obviously, it's a genetic disorder. It's 100% heritable. But given that we can modify the environment, after environmental modification, is PKU in itself still genetic? And you can think about this with respect to exposures to many, many different things, including, certain types of diets and so on and so forth. There's even anecdote of studies where part of a population has adopted say a traditional diet, and another part of a population has adopted a new diet. And in those populations in adolescence, height doesn't look heritable, or body size doesn't look heritable. And we know that those are, among our most studied traits throughout history, and we know that those are heritable. But when you induce environmental variability, sometimes you see the greater effect of genes. And so this has obvious applicability to substance use disorder. Who has substance use disorder absent exposure to a substance? Well, nobody does, right? But that doesn't modify an individual's genetic risk for a substance use disorder. The genetic risk is fixed, but it's contingent on the exposure. And so far, I've given you lots of examples as to why you may think, well, substance use disorders are really difficult to study, because there's this long list of potential symptoms, and then some combinations of them lead to diagnoses. And there are these stages of the disease, and without exposure, well, because we can study preclinical models, and because we've been interested in this topic for centuries, much is actually known, and there is a basic neurobiology for understanding how substance use disorders develop. I'm not going to go through this, but this is a highly cited paper that describes a theory for the cycle between preoccupation or anticipation of use, use that results, or a binge that results in intoxication, subsequent withdrawal and negative affect from ceasing use of that substance, and then subsequent preoccupation and anticipation of use again. And that's the cycle that happens. And the neuroscience of this is much of it is worked out well in preclinical models and more in human models. And it may then seem obvious that, well, if we understand this basic neurobiology, we understand the genes that contribute to contribute to these biological pathways, and maybe we ought to just focus on studying those. And I'm sure many of you are aware of the candidate gene era in human genetics, where as the Human Genome Project progressed and genes were identified, many of those genes that were involved in these processes, for instance, neurotransmitter receptors, like this idea that most psychiatric disorders are disorders of dopamine, and let's study the dopamine transporter and every dopamine receptor. We've been through that, and unfortunately, not with a great deal of success. So although we know much about the biology, the presence of variation in genes that meaningfully impact function in living adults and lead to variation in risk for developing a substance use disorder after use, that's the bridge that you have to cross to get from understanding the neurobiology, that it's meaningful there, and understanding what genes are in it. But that variation in those genes, that important variation gene in those genes, has to be there for us to see the impact at the population level. I just want to talk a little bit about how we define cases and controls. In genetics research now, I'm sure you're aware, we are, in human genetics research, we're frequently now using very, very large samples gotten at through massive population-based efforts like All of Us in the US, UK Biobank in the UK, some population-based samples in Iceland or other Scandinavian countries, some big electronic health records, biorepositories in the US. And so frequently in our largest genome-wide association studies, we aren't relying on cases that we go out and get and controls that we go out and get. And so we really have to start thinking about, if we're doing a specific case control analysis, what is a case and what is a control, and how defining cases and controls will impact the interpretability of the findings. So if I use as controls individuals who've never used a substance, and cases people who have a severe use disorder, I may be unintentionally studying a process early on, which is the transition from non-use to use, right? Because the thing that distinguishes those two groups is that one has used substances and the other hasn't. That's probably has more to do with personality or psychological constructs like novelty seeking, environmental factors like drug availability, parental supervision, whether or not someone initiates use. Others have taken the approach to defining controls as someone who has used enough substance to potentially develop a use disorder, but didn't develop a use disorder, and cases as people with a use disorder. So for instance, someone who smoked 100 lifetime cigarettes, but never become dependent on nicotine. Someone who's used opioids 10 times, but never developed a use disorder, versus someone who developed an opioid use disorder. And so if you think about that, the difference in those cases versus controls, they've all experienced the exposure. So think back to the PKU example, they've all experienced the exposure. Some developed a use disorder, some didn't. And so clearly what you're studying there are differences in pharmacology, in pharmacogenomics, for instance, an or not, but an individual's response to a substance. So these are clearly different traits, right? One of them is early behavioral and environmental. The other is clearly probably a more biological phenotype. And so it's just important to think about when we're studying substance use disorders, what we're actually studying. And probably the best way to think about substance use disorders in the population and the genetic contributions and the environmental contributions is to consider a continuous underlying liability that's contributed to by many genes. I'm sure you're familiar with Fisher's work, but this is a Fisherian perspective on human disease. And so in the population, there's some mean of the liability, Again, the liability isn't just genetic, it's contributed to by both genes and environment. And as we move along that liability distribution, above some threshold, everyone or some increased probability of individuals are affected with that outcome. And so, as we move to the right on this normal distribution, people to the far right are much more likely to develop a disorder. But in reality, liability to substance use disorder, for instance, or you can imagine that this would apply to type 2 diabetes, obesity, a lot of other what people might call lifestyle disorders, that over time, an individual at time zero is inherits some initial liability, or even at conception, and then through the prenatal period that can move based on the prenatal experience, and throughout life, an individual's liability can move to the left or right based on their experiences, epigenetic change. For substance use disorder, anything that increases the likelihood that someone will use a substance is going to shift them to the right. Anything that decreases the likelihood they're going to use will shift them to the left. And at whatever age we decide we're going to study someone, that's where we're measuring their phenotypic outcome. Okay, so let's talk about this. Why, the history of human genetics I'm in a school of public health, but the history of human genetics in public health has not always been positive, and but we're at a point now where information is available. We are beginning to understand how an individual's genomic makeup impacts their risk for certain outcomes. Moreover, we're now starting to understand the pharmacogenomics in a way that we could inform treatment, and within substance use disorders, we may be able to inform medication-assisted treatment. But the reality that we're faced with now is that millions of citizens already have access to their genetic and genomic information through commercial companies like 23andMe, and their risk for particular outcomes is being communicated to them without the presence of someone like a genetic counselor who can help them better understand that risk. In humans, we estimate heritabilities usually from twin studies. This is the range of heritabilities from a meta-analysis or meta-analytic review more than about 20 years ago. Twins, there haven't been a lot of twin studies in the last 20 years, so the heritabilities might change a bit. But you can see the heritabilities range from a low of 0.38-ish up to a high of 0.7-ish. The order of this doesn't really follow any particular class or category, but you can see that there's a substantial contribution of genes to the risk for developing a substance use disorder. Subsequent studies have shown that there are separate latent genetic factors, one for illicit substances and one for illicit substances, which is an interesting notion. And you'll note there that cannabis is on the is in illicit category at this point in time. It'll be interesting to see if there are large enough twin studies to look at this, how that changes over time if cannabis dependent symptoms move to the illicit side of that. But they're highly correlated, those two latent genetic factors. So the one important point is this isn't gene finding, right? This is just using a statistical model within individuals who we know their degree of relationship and estimating the relative contribution of genes. Given what you all do, I think you're probably pretty familiar with these kinds of approaches, but this doesn't get us to actually understanding the genes. I've already mentioned this concept of common liability. It is interesting that substance use disorders are diagnosed specific to particular substances. So people have an alcohol use disorder or a cannabis use disorder or a cocaine use disorder. They're not diagnosed with a general substance use disorder. But we do know that based on the previous slide and some other seminal work by Ken Kendler and his group, and Ming Shuang earlier in his group, have shown that that they can estimate in a twin model that an increased liability for one substance is correlated with an increased liability to develop a use disorder for another substance. And the way to interpret this is that there's a common set of genes that are increasing the liability to any substance use disorder. And then there are drug class specific genes that are contributing to the risk for developing a particular substance use disorder. So a common set of genes contribute to alcohol use disorder and cannabis use disorder and nicotine dependence and an opioid use disorder. But then there are some drugs that are uniquely contributing to the liability to develop a specific drug substance use disorder. And so in general, things that impact externalizing behaviors like ADHD related behaviors or conduct disorder related behaviors or novelty seeking behaviors are probably among the common genes that are contributing, as well as genes that are involved in pleasure, like in the limbic system, may be common genetic factors in influencing substance use disorder overall. But enzymes, for instance, that are in specific metabolic pathways for a drug class are probably modifying the likelihood of developing a particular substance use disorder. So, when someone smokes, variation in their nicotinic receptors may impact reward that they feel, the amount of reward that they feel when they smoke, and that's going to have a specific effect on developing nicotine dependence. Genes that are involved in alcohol metabolism are known to have a pretty big impact on the risk for developing an alcohol use disorder. And this is especially apparent in the population because the exposure to alcohol is so frequent. And then the other thing is, we don't expect these genes to have the same impact over the course of the disease, right? So the things that are impacting environment aren't the same, the things that are impacting initiation are not the same things that are impacting the transition from abuse to dependence or from dependence to cessation, right? So it's a pretty complicated set of questions. So it's a multi-stage disorder with an obligate exposure, an underlying biology that is itself impacted by the disorder stage. We have a neurobiological model that's somewhat accepted. We have a dimensional definition that fits well within a liability framework. The liability is reflected by the disorder stage itself. So this requires multiple investigative approaches to understand the relative genomic and environmental contributions across the course of the disease. And unfortunately, I'm going to show you study findings, and I'm trying to think of any exceptions, but I don't know that any of them actually accounted for any important environmental factors. But over time, we'll get better at that. I mean, that's one of the things. The first, the early era of big GWAS in psychiatric and substance use disorders largely ignored environment. And then I think as we move forward, as the studies get bigger and we start to get a handle on what the top findings mean, I think we'll worry more about the interplay between the genome-wide genes and the environment. So to date, there are multiple replicating findings from genome-wide association studies, or GWAS, for substance use disorders. Largely, these are the result of really large consortia. The biggest consortium is the biggest of these is the Psychiatric Genomics Consortium. This has done work on schizophrenia initially, and then every other psychiatric disorder since. There is a specific substance use disorder group which has brought together collaborators from around the world to combine their samples. I think for substance use disorder, I know for substance use disorder, the largest publications to date have millions of individual subjects in them. So millions of people have genome-wide genotyping done and are assessed on at least one substance use disorder. And the findings we have so far are the specific replicating findings are drug class specific and in metabolic pathways. So for instance, nicotine dependence, nicotinic receptors, alcohol use disorder, aldehyde and alcohol dehydrogenases, caffeine dependence, cytochrome P450 enzymes. And there are emerging successes for opioid use disorder and cannabis use disorder. And again, these are genes that probably would be candidate genes if we knew the biology, if we had known the biology. So they sort of make sense when they arise from these studies. This is a work that I was part of. This was the first cross ancestry, meaning that we combined subjects of European ancestry and African American ancestry and Asian ancestry together to perform a genome-wide association study of alcohol dependence. And there are these, maybe just in case you're not familiar, but this plot here is a Manhattan plot. Along the x-axis, it's the chromosome, and from one terminal to the other, the base pair position. And each dot represents the minus log 10 of the p-value of an association test between an allele at that SNP and the outcome, in this case, alcohol dependence. And you can see there's one signal that's, 1 times 10 to the minus 10, 0.0 and 9 zeros and a 1, very significant finding. And that is in ADH1B, which is among the genes that I just mentioned in the metabolic pathway for alcohol. More recently, another large consortium that I was part of looked at cannabis use disorder, and you can see that with 20,000 plus cases and 360,000 plus controls, there were 2 what we deem genome-wide significant. That means significant after correction for performing millions of tests. This isn't really the interesting finding from that study. This is the interesting finding from that study, and that is that given that we now can create sort of polygenic risks, or I'll talk more about them, but understand the polygenic architecture, like the joint contribution of a whole bunch of genes to the risk for something, we can start to ask questions. Does the genetic risk for trait X correlate with the genetic risk for trait Y? And in this, we looked at cannabis use and cannabis use disorder and its correlation with the genetics of other traits. And you can see, for instance, that it's significantly positively and negatively correlated if it's use or use disorder with body mass index and so on. You can see there's a significant genetic correlation between use and use disorder pretty much across psychiatric disorders. So the green ones are psychiatric disorders: anorexia, ADHD, bipolar, major depressive disorder, post-traumatic stress, and schizophrenia. And then you can see with other use disorders, those are the red or pink ones down below. It is interesting, there's a lot of work now on understanding the causal relationship between the genetic risk for cannabis use disorder, cannabis use itself, and the development of schizophrenia. This is, I think people now call this pleiotropy. I think in the past we thought of pleiotropy as like one SNP influencing 2 traits. Now, now we have a better understanding that there are aggregates of genetic risk that are correlated across phenotypes. So it's interesting. Polygenic risk, the concept of polygenic risk. I don't know how much this is applied with livestock now, but in humans now, this is sort of like our approach du jour for the last 10 to 15 years has been to take the top results from GWAS and create in a separate data set, create weighted sum scores. So the number of risk alleles that a person has weighted by that alleles, the log of the odds ratio for that particular allele, and sum that up across the genome. And that gives us an idea of an individual's polygenic risk. And so you can see like this cartoon example here of cases and controls for the best genetic polygenic score. You can see that there's cases and controls separate nicely. It doesn't always work out so well, but there are examples like this one here from this paper. I was part of this also, the schizophrenia working group of the Psychiatric Genomics Consortium. You can see that on the lower right panel, as we move up in decile, this is 3 different cohorts, but in the top decile, the odds ratio for the risk of developing schizophrenia among people in the top decile was a 20-fold increased risk over the lowest decile. And in the sample with the weakest association, it was a 7-fold risk. So this is kind of important. Schizophrenia has a population prevalence of, say, 1-ish%. In one of these samples, and the people in the top decile had a prevalence of, say, 18% or 17%. And so this is probably clinically meaningful. This paper was published about 10 years ago. And to date, we don't apply polygenic scores in the clinic for schizophrenia. Let me talk a little bit about some of the applied polygenic analyses we've done in Baltimore. So colleagues of mine have this center called the Prevention Intervention Research Center, and they've developed ascertained a couple of cohorts that they enrolled in kindergarten or first grade and followed into adulthood. And about 1,350 of those subjects were ultimately genotyped, and we have assessments of substance use and a whole bunch of other traits. And this is just a sampling of the probably 20 papers we've now published looking at the association between polygenic risk for a particular health outcome and how it impacts development in these kids from kindergarten into adulthood. I think an important point here is, although we get statistical significance in all of these cases, the magnitude of these findings, we're talking about variance accounted for in the single digits. This isn't like 10% of the variance accounted for, 20% of the variance accounted for. This is like 2-3% of the variance accounted for. And this is pretty common in human genetics now, seeing these kinds of findings. So just a brief summary about polygenic scores. They're useful for translating large-scale study findings to measurable indicators of individual risk, but it is very premature to think that in most cases for psychiatric outcomes, for substance use outcomes, that we're even close to being able to apply these clinically. That said, if you use Ancestry.com or 23andMe, you log in now and you can subscribe to a higher level of the service, and you can see your polygenic risk, usually in like quartiles for a whole bunch of outcomes, usually not disease outcomes, but like, will you like to dance or things like that. And I can promise you that the variance accounted for by any one of these is always in the single digits. And so it's interesting as an adult to read like, oh, it says that I would enjoy dancing, and you already know whether or not you enjoy dancing. So, okay, so let me move on to epigenetics and addiction. So, sort of the hypothesis is that someone would get exposed to drugs, and based on this active drug exposure, there will be epigenetic changes, and those epigenetic changes will modify function of the genes, probably through changes in methylation in the promoter region of the genes, and that will up or down regulate the gene's expression. There are other epigenetic changes too that can happen. And our thought is that part of that process, that cycle that I showed earlier in the worked out model of the neurobiology of addiction between use and then withdrawal and then anticipation of use and then withdrawal and anticipation of use, part of that, what causes that cycle to happen among people who are frequently drug exposed, is epigenetic change that leads to that anticipation of the subsequent use. And so much work that colleagues that I'm working on with colleagues now is on understanding this process, studying animal models, studying the brains of individuals who had a use disorder post-mortem. They had a use disorder, and now we've gotten post-mortem tissue, but also in peripheral tissue sampled from people who are using substances over time. And the obvious thought is that if we can understand, if we look across the genome and we highlight and we find differences in particular genes in methylation between users and non-users, that will highlight pathways where epigenetic change has happened as a consequence of use, and that may highlight pathways which are part of maintaining the substance dependence or substance use disorder process, and that if we can intervene in those biological pathways, and then with whatever approach, but pharmacotherapeutics, for instance, that we could disrupt this. So DNA methylation typically refers to comparing the proportion of cytosines that are methylated at specific sites between two groups. You could also do a continuous measure of methylation versus a continuous outcome. Methylation could be represented as individual CPGs, groups of neighboring CPGs like CPG islands, or global levels of DNA methylation calculated using some kind of an algorithm. And now we are frequently using DNA methylation aging approaches to approximate someone's biological age instead of their, or relative to their chronological age. The outcomes of interest could be disease status, environmental measures. Remember, this is a means of measuring the impact of an environment. Quantitative traits, genotypes. So we could look at the relationship between methylation and the genotype and understand that sometimes methylation levels are actually genetic. Or gene expression levels. How is it that the methylation differences at particular genes impact that gene's expression? And methylation can be measured multiple ways, but the two main approaches are using array-based approaches and sequencing-based approaches. So there were some early great successes looking at substance exposure and DNA methylation changes. This is DNA methylation in newborns and maternal smoking during pregnancy. This is the result of a consortium meta-analysis. And you can see there's like a 1 × 10⁻190 or something, but 6,073 CPGs were considered statistically significant. So you can see that the black lines down low, everything north of that is considered statistically significant. And I think this was a 450,000 or 27,000. There's probably 450,000 sites. So you can see a massive number were statistically significant. So that gave us hope that, okay, if we can look at people who've been exposed to drug, then it might help us. If we can look at people who've been exposed to drug, we may see methylation differences between people who have been exposed to drugs versus not, or people who are actively using drugs versus in periods of cessation, the same individuals. If we look at those subjects, we may see methylation differences that aggregate in particular biological pathways that help to provide us with information about the biology of the use disorder. And so let me briefly introduce the AIDS linked to the intravenous experience, or a live cohort. The PIs are my colleagues Shruti Mehta and Greg Kirk. It's a prospective cohort study designed to characterize the incidence and natural history of HIV infection among people who inject drugs in Baltimore. Over 5,000 people have been enrolled since 1998, and assessment happens twice annually with biosampling. We have genome-wide SNP data, or single nucleotide polymorphism data, on now over 3,000 subjects, or about to have it on about 3,600 subjects. But we have genome-wide methylation data on about 450 subjects during 1,200 visits, so repeated measures on the same individuals. And I picked those individuals based on whether or not they were actively injecting drugs versus abstaining from all drugs of abuse or recreational drugs. And this was using peripheral blood. And we had longitudinal drug use data on the subject, so we could verify that. But it's, again, self-reported data. This is just a table of the study characteristics. You can see we had 397 observations when they were not injecting and 390 where they were. We reduced this to paired visits per subjects. And this first analysis we did was for HIV infection. And you can see for HIV infection, we see a bunch of significant results. And so what this tells us is, wow, for HIV infection, there are pretty significant epigenetic differences that we can detect in peripheral blood. Not a huge surprise, but it provides sort of a positive control for us. Like if we're looking at this thing that's clearly going to have a biological influence and an impact on someone's health, HIV is pretty good. So if we look at those with and versus those without HIV, we can see that we get really nice results. And this gene NLRC5 is located on chromosome 16q13. It is interesting because the virus itself integrates into specific parts of the human genome, and it is known that NLRC5 is one of the places where it actually integrates, and so it's likely that it's just disrupting methylation or changing methylation patterns where it's integrating into the genome. And so the question is this non-random integration, or does this integration here impact disease course? And actually an interesting question is, does where HIV integrate in the genome itself impact it? We also ran an epigenome-wide association scan to look for methylation differences at specific sites throughout the genome. And just to step back, we used an array of 850,000 sites. So we're getting 850,000 sites throughout the genome. It's an Illumina product, the Epic chip. And on the upper panel, the A panel, it's a QQ plot. And so this is the expected versus observed test statistics. And we can see that because the line is pulling away from a slope of 1, we see a significant enrichment in signal. But when we look to the right, we can see that only one site is actually genome-wide significant. With cocaine, it looks a little bit less enriched, and you should note the differences in the y-axes here. None of these are genome-wide significant. So it's not like HIV infection where the biology that's being highlighted is very clear. We can look at the overlap in the methylation sites that were significant by type of analysis we did. So we did analyses for any injection drug use, for frequency of injection drug use, for use of heroin, for use of cocaine by injection. And you can see in the center here, like, I don't know, 24 CPG sites overlap everything. 24 genes overlap everything. But we did a lot of follow-up work to see if this was highlighting particular pathways. Because this is injection use, a lot of the pathways that were being highlighted were those that are involved in like inflammation and immune response. And probably what it is just the body's response to chronic injection is what we were seeing there, and not really like drug class specific stuff. So some more work needs to be done to tease this apart. And so we have those things going on. The one very interesting finding that was consistent across all of our analyses was this gene called FKBP5. It's a particular like Hopkins interest in FKBP5. It's located on chromosome 6. There's a huge amount of existing evidence on the role of it in stress response and of differential methylation in response to chronic stress and stress-related psychiatric disorders. So probably the best way to interpret this is that the chronic injection is eliciting a stress response. And so we're seeing differences based on current injection state and methylation in this. A lot of people have thought of this as like the degree of methylation at FKBP5 as like a barometer of stress response. Some other work we've done recently, and this is ongoing work. We're not part of a large consortium. We have a meta-analysis that's on medRxiv, so it's a preprint that's available publicly and is under review at a journal now, that combines 4 existing cohorts beyond this one to get at results. But interestingly, the top finding we have from this one cohort that we did locally replicates across the other samples. So this was in collaboration with something called the Lieber Institute for Brain Development, which is a Hopkins-affiliated independent research institute. We generated data from the dorsolateral prefrontal cortex. It isn't single cell, it's bulk tissue. We're able to estimate the percent of the sample that's neurons and account for that in our downstream analyses. But then from the bulk tissue data, we generated genome-wide methylation data using the same chip that we used, or the same array that we used in the ALIVE cohort. And then we also generated RNA sequence data to examine case control differences in gene expression. And I'll show you, this is what the sample looks like. One thing we did is we had among our controls, we tried to group match based on psychiatric disorder. Many people with a substance use disorder will also have a psychiatric disorder. And so we included psychiatric controls, people who have a psychiatric disorder but not a substance use disorder. This is our methylation study. Nothing is genome-wide significant. So across 850,000 sites in a relatively small sample, but big for postmortem brain work, we didn't find anything genome-wide significant. But when we looked at the RNA sequence analyses, we find a gene that was significantly underexpressed in the subjects who died of an opioid overdose. And so just to step back to what this cohort was, these are individuals who have a positive tox screen for opioids at the time of death, and the medical examiner determined that their cause of death was acute opioid intoxication. Our control sample is age, sex, and race group matched, psychiatric disorder group matched, but did not were tox screen negative for opioids at the time of death. And also, there's a family informant interview post-mortem, and based on the family informant interview, none of the folks have a substance use disorder. And so this NPAS4 becomes an interesting gene. It's on chromosome 11. It encodes for neuronal pass domain protein 4. It's an immediate early gene, which is a transcription factor, which is a gateway to transcription of other genes. There's a host of rodent model data on this that it's differentially expressed with reward-related behaviors, including acute cocaine and opioid administration. I mentioned this already, but just in, we were putting together a consortium center on studying opioid genomics, and among the other cohorts, when they did a lookup for this gene, it was nominally significant, meaning a p-value less than 0.05, I think, in all of them, but it was in the near the top of the list for 2 of 3 that we immediately looked at. But formal meta-analysis has confirmed that this is associated with opioid overdose risk. And so what next? Substance use disorder is hard to study for a variety of reasons. In our ongoing populations, we are developing approaches to measuring using EMA, or ecological momentary assessment, measuring in real time someone's substance using experience. So for instance, when's the last time they used, how much they're craving the substance, how they're, whether or not they plan to use, things like that. We also have people where we're working with, collaborators who are developing wearable devices that may give us a sense of an individual's physiology while they're using. We're working on assessing other biomarkers that may be better markers of risk for other substance use disorder outcomes. Obviously, especially for the postmortem brain work, larger samples for replication and signal refinement. And then the hardest thing, and like I mentioned, I'm part of a consortium that's working on this, is integration across domains, biological domains, and integration across organisms. Again, I've mentioned this multiple times, but there's a host of data, massive amount of data in preclinical settings. For instance, rodent work, rats and mice, on, brain gene expression patterns after acute exposure, after chronic exposure, and bringing that to studying both in humans and then to clinical models has been a challenge, but this is sort of where the field is hopefully heading. Okay, so conclusions. Twin studies indicate a higher heritability for substance use disorders. Many of the genetic influences on substance use disorders are shared across drug classes. Liability is this idea of a continuous risk for a substance use disorder is a useful metric for capturing the dynamic nature of substance use disorder risk. And the nice thing about it is it captures everything: behavioral, environmental, and biological dimensions of risk, and it could do that in one indicator or one metric. The nice thing is it can also capture protection or resistance or resilience, right? So it doesn't just have to capture risk. Large genome-wide association studies are starting to bear fruit. Exploiting the polygenic architecture of substance use disorder-related phenotypes allows examination of genetic correlation between traits. As I showed you, cannabis use disorder genetic risk is associated with risk for a bunch of psychiatric disorders and risk for other substance use disorders. Epigenetic influences are likely to play a very key role in the addiction process after drug exposure. And I think probably this is where we'll learn the most important components of the biology that will inform the development of downstream treatments. But to date, larger samples and replication are needed, especially for in epigenetics. And literally, my next call is to work through writing the aims for a grant to support an international consortium to integrate across multiple tissue types in humans and multiple substance types epigenetic data to hopefully get to this bigger goal. And this is just, some of my collaborators over the years working on the things that I've talked about. And that's it. And I'm going to end the slideshow. And it looks like there's some time for questions.
Yeah, as we have. Thank you for the presentation. Was amazing. It's always nice to hear from people from another area of expertise.
Yeah,
I
for
think
sure.
It brings a new perspective. Yeah. So
yeah,
if
and it's been
amazing
for me. I've a lot of my collaborators now are preclinical in the preclinical space and do work with rodents, and it's amazing to have a conversation to think about the different approaches. Like right now, we're developing an animal model for overdose risk, a mouse model for overdose risk. That's interesting, right? And something we can, as a trained in human genetics, something you would never even, dream of as possible. But like, a high, respiratory depression in response to standard doses of opioids is a pretty good proxy for overdose risk, things like that.
Very, very interesting. If anyone has questions, you can raise your hands, turn on your camera and microphones. Yeah, so we do have a question here in the room. Bruno, you can go.
Okay, first of all, congratulations for your lecture. It was very, very good. Here's Bruno. I would like to ask you if you have observed any correlation between pregnant mothers' use or abuse of drugs and offspring methylation.
Yeah, there's actually a paper, several papers, and I should be an author on one that's published and one or two that are in the pipeline from the the Boston birth cohort. And so the first author of those is someone named Henri, Henri, Dessany, Dessany. So we see methylation differences in cord blood. The interesting thing is that one thing I presented was in the newborns, right? And there have been follow-up studies that some of the methylation changes, newborn exposure to tobacco persist into early childhood. And this is of particular interest. I get the question you're asking. This is of particular interest because I'm also interested in whether transgenerational effects are there and whether or not a mechanism by which transgenerational effects happen is due to prenatal exposure. But yeah, they exist, and multiple studies have seen persistent methylation differences in the offspring of mothers who were using while pregnant.
Thank you.
Mm-hmm. I think Michael has a question.
Yes, thank you for a really informative and fascinating talk. You might not be surprised that in animals, we're also interested in epigenetics. And I'm just curious, because some of your comments at the end talked about how some of these effects are epigenetic or can become epigenetic. I'm just wondering, is there how reversible are epigenetic effects once those take hold?
Yeah, isn't that like the $1 million question right now? Isn't that the company that was started, I don't know, the founders, which billionaires started a company and gave multiple folks who study the epigenetics of aging 7-figure salaries to come and figure this out. So the fact that I didn't get the 7-figure salary would tell you that I don't know the answer yet. But We know, for instance, that the whole classes of drugs methylate and demethylate, right? So the important point, and I don't know the details of this, but I just saw a report of using CRISPR-Cas9 machinery to actually change site-specific methylation. So really, it isn't like, can you methylate, can you demethylate? It's can you target it in a way that's meaningful, right? Because if you just do like blanket global methylation, demethylation approaches, there are going to be so many off-target effects that it's bad, right? And then it also begs the question, like, at what point in humans will we do that? But CRISPR-Cas9 gene therapies are already in the market, right? These classes of drugs that are incredibly expensive, like 6-7 figures per dose, including for sickle cell disease. Those are tissue-directed CRISPR gene editing. So I would think that we're going to be able to do the same thing with modifying methylation in the not too distant future. But again, as you know, it's got to be the right tissue, the right cell. It's just not going to work for everything. But yeah, certainly methylation can move in either direction, but the ways to make it happen in a organized and site-directed way are the difficulty. So I think yeah, sure. I think Flavio has a question.
Yes, I have a question.
Sure.
Excellent talk. Really,
Oh,
really
thanks.
nice to hear
Thanks.
from another field. I'm a
Yes.
professor here in the work lot to genomics, but livestock. Bruno asked a question that I would ask about these transgenerational effects, but I have two more. The first one is more like I'm curious because I didn't read your papers, but which method are you using for the GWAS analysis?
Which method are we using? So
yeah, for GWAS.
Yeah, so we genotype by array. None of that's genotyping by sequencing. We typically impute different panels to a common reference panel, like 1,000 genomes or TopMed.
Yeah.
And then the approaches are usually linear regression or logistic regression, including a couple of covariates. The covariates that we always include are those that represent population structure, which is like the genetic reflection of a bunch of things that we call race, ethnicity, ancestry, shared common history. And those Manhattan plots represent the -log10 p-values that we get out of the additive effect of the genotypes at one location.
Yeah, so like the allele substitution effect. But I mean, the method you treat as a random effect, the markers and fit all markers at the same time, and them all fit one by one.
Yeah.
And then you have some variables to correct for stratification. But did you include the polygenic effect? In other words, the genomic relationship matrix of the subjects?
So some of that happens here and there, but that's not the standard approach that people take. But I agree with you. You tend to know the degree of relationships between your subjects and your studies, and they tend to be more closely related. And so when we have people, for instance, people from within families, we'll use a mixed model or something like that to account for the degree of relationship. But in most studies, it's just principal components of ancestry that get included to account for the differences in the degrees of relationship between individuals.
Yeah, because mostly the subjects are not
not
closely related.
Yeah, they're not closely related. Right.
Yeah, that's okay. And my second question is, when you define the phenotype, you are defining a binary trait, right?
Yeah.
So we a single threshold. But when I look at the description, right, of the transition from one state to the other one, that's the disease, would not be worthwhile to have an intermediate phenotype, right? That would be kind of one that is starting to use the drugs. Is not in the already dependent, and that you would say with the disease. So my question is, probably there is a study that you did compared if you like a phenotype that have a multinomial distribution, that have more classes, would not be better than just say 2.
Yeah. Yeah, I agree with you. The question, so it's always this difficulty in, so there's always this pull and push between what clinicians think is a reasonable metric for something, what our funders think is a reasonable metric, and then what theoretically we think is a reasonable metric. And unfortunately, for the really, really big studies, we just don't have subclinical phenotype measured, right? We have like, the obvious outcome versus, obvious absence of outcome. But longitudinal studies that follow the entire course of someone's drug use, like that's probably the best way to get at it. And we're hoping that we'll have those in time, over time, to get at like degrees of severity of substance use disorder.
Yes, yeah, that's what was
Yeah, it
thinking,
would
right?
be,
To have at least one intermediate case, right, where the person is not in the severe case of dependency, right?
Yeah.
Yeah.
Yeah, ideally we could study that.
Yeah.
Ideally we could study it.
Yeah, because when we have that liability scale and then you transform to a binary trait, you lose information, right?
Yep.
If you could transform to, well, great talk. Thanks for doing
Yeah,
this.
thank you for inviting
Very,
me.
very,
Yeah, I
very
really
nice.
enjoyed it.
I'll
Awesome.
see if others have a question, but thanks a lot.
Yeah, and I'm sorry, I do have to run. I have another call, unfortunately. I can't hear you. I can't hear you.
Something happened there.
Can
Okay, nice.
you hear me
go.
now?
She's good.
I can hear
Yeah.
you. Yeah, yeah, yeah.
Okay. Well, we appreciate your time, and
Yeah.
your talk was very good, and we hope to have you again in the future at some point.
Yeah, definitely invite me again. I'll talk about something different, something newer next time. We have a whole bunch of stuff.
Okay.
We've sorted cells we're starting to do single cell homogeneous cell stuff. So that would be interesting to talk about in the future.
Yeah, absolutely. Okay, thank you.
Bye-bye.
Bye.